Hi, my name is Mark McPeak, and I'm a professor in the Biological Sciences Department at Dartmouth College. And I'm going to be talking to you today about the ecology of infectious disease dynamics. Let me see here. I've never given a lecture sitting in the cushy chair in my house, so we'll see how this goes. Hopefully we'll have some fun with it. And hopefully I can teach you a couple things. So as I said, we're going to be talking about the ecological dynamics of infectious diseases, or you can think of this as the interaction between two species, a pathogen and its host. So this is what we're going to talk about today, is really the first inklings of how different species interact with one another. And you're going to get a lot in this class about this. Now, before you watch this, I would like you to read these two papers. Paper by Litvinova et al. From last year, that is the reactive school closure weakens the network of social interactions and reduces the spread of influenza in the Proceedings of the National Academy. And another paper by Catherine Glass and Belinda Barnes that is older, in 2007, on how much would closing schools reduce transmission during the influenza pandemic. So what I'd like you to do is read these two papers, think about what they say, and then come back to the lecture here and watch the lecture. And then at the end of watching the lecture, go back and read the papers again and see if you have developed any more comprehension about the concepts we're talking about and the way people think about disease dynamics in populations. Okay, let's get started. So we're going to start this story not talking about a model, but actually talking about this fellow, Sir Ronald Ross. Sir Ronald Ross won the Nobel Prize in Physiology or Medicine in 1902. And he won it because he is the person who proved that mosquitoes are the vector of malaria among humans. So that malaria is passed among humans by a mosquito biting one individual and then transferring pathogens to the next individual it bites. It's a very interesting story. So Ross actually made this discovery. He started doing a series of experiments in 1894 where he looked at the life cycle of the mosquito and tried to figure out where the malaria parasite was entering this life cycle of it. And it was also very telling because Ross did a series of experiments. He didn't rely on just observations. He actually did experiments. Some of them we would think of as kind of questionable today, maybe, but he did them, and this is how he proved it. What he did was he paid a local man in India, because he was stationed as a doctor in the British Army during the Raj period in India, and malaria is very prevalent in India. So what he did was he raised mosquito larvae from eggs and then allowed them to merge in the lab so he knew that those mosquitoes were not infected with the malaria parasite. Then he took them in a jar and put them on a local Indian man that he would paid for every mosquito the man would let bite him in his jar. He then took the mosquitoes back and let them grow for a few days on the blood meal that they had taken. And then he killed the mosquitoes and dissected them and looked at their guts to see if he could see the parasite. And in fact, he did see signs of the parasite. So this is a page from his scientific notebook for August the 20th and 21st in 1897, when he described what he saw as pigmented bodies in the gut of the mosquitoes that he dissected. So here are his little hand drawings of the little guts that he saw in the mosquitoes, and these little dots are what he saw as the malaria parasites being growing in the gut of the mosquito. This was revolutionary because now physicians they knew how to stop malaria in humans. If they could decrease the mosquito population or prevent mosquitoes from biting individuals, you would slow down the rate of transmission of malaria among humans. So let's take a little sidetrack here for a second, because this is a biology course. We're going to talk mainly about math, but let's talk a little biology. So what causes malaria? Malaria is caused by a pathogen that is actually a protozoan. So it is a eukaryote, just like you and I are. And it's in the phylum Apicomplexan. And it's so devastating to humans because what Plasmodium parasites do is they want to live in red blood cells. So they infect red blood cells in your bloodstream if you're infected with malaria, and they basically make the blood cells pop. So you have fewer red blood cells. The red blood cells have more problems passing through capillaries. As those red blood cells are circulated through your circulatory system, they cause all kinds of problems. Well, malaria, where did it come from? Malaria, like many infectious diseases in all organisms, are typically caused by jumps between species that serve as the hosts for the parasite. So in the genus Plasmodium, there are over 175 species that have been described in this one genus. There are 4 that infect humans: Plasmodium vivax, Plasmodium malariae, Plasmodium ovale, and Plasmodium falciparvum. Now, these, as well as others, Plasmodium species infect at least 29 different non-human primates worldwide. And they infect rodents outside tropical parts of Africa, but they only do this rarely. But lots of other mammals are not infected by them. The other main hosts of malaria parasites are birds. And in fact, if you read some of the literature, bird populations, all the endemic birds on Hawaii, many of them have gone extinct, and a lot of the species have collapsed, the native species of birds, when malaria was introduced in the late 1700s and early 1800s to the Hawaiian Islands. Plasmodium vivax and Plasmodium falciparvum are the most common plasmodia that infect humans, and falciparvum is the parasite that causes the most damage in humans, and that's the most deadly of the 4 that infect humans. But falciparvum, the reason malaria is such a problem in Africa is falciparvum is the primary malaria parasite in sub-Saharan Africa. Now, what does a parasite do? Most parasites have very complex life cycles. So this is just a diagram of what the malaria parasite does in the mosquito, when it's living in mosquitoes, and then when it is living in the host of humans. It's a very complex system, and if you would take a class in parasitology, basically you'd spend all your time sort of learning these very complex life cycle graphs of the parasite. But what this does is it gives you some idea about where the problems for symptoms come in humans when they're infected with parasites, but they also, by understanding these diagrams and the life cycle history, you can get an idea about where it may be best to attack the parasite in the stages that it works on. Now, for malaria, there's been a coevolution between malaria and humans. And some of you may know this, but there are lots of human populations that actually have adaptations to living in an environment where malaria parasites are prevalent. One of them is sickle cell anemia. So sickle cell anemia is caused by an alternative allele at a hemoglobin molecule, the hemoglobin molecules in your red blood cells that carry oxygen through your body. Sickle cell anemia is actually an adaptation for living with the malaria parasite. We're not going to get into that in this course or right now, but if you're interested more in this, you should take my evolutionary medicine class, Bio 48, where we'll talk about the coevolution of hosts and pathogens. Sort of the evidence for that is that if you look at the frequency of the sickle cell allele in human populations, there's a very striking co-relationship between the frequency of sickle cell hemoglobins and the frequency of malaria parasites. So this map shows you the distribution of falciparum malaria across Africa and Southern Europe and Southern Asia. And this is a heat map that shows you the frequency of sickle cell alleles in those human populations in those same areas. But let's get back to Sir Ronald Ross. So Ross made this discovery, won the Nobel Prize in 1902, and then went back to England to take up practice in England there as a research physician. He enters our story today because Ross really published the very first mathematical model of the dynamics of interactions between two species. So we usually, in this course, you're going to talk a lot about interactions between predators and prey, competitors for resources, apparent competitors, food webs, all kinds of things. But the very first model of one species interacting with another species and how they influence one another's dynamics was actually this model by Ross, which was the description of humans interacting with a malaria parasite. This was published in Nature in 1911. And as I said, it was the very first paper to describe a model of an interaction between two species. The next year, Alfred Lotka published a note on Ross's paper, also in Nature. This is the entire paper right here in front of you. So if you really want to read it, right there it is. And what he did was extend Ross's analysis, because Lotka was a very famous mathematician, and he presented a solution to Ross's system of equations that he had set up. Now, you're going to hear a lot more about Alfred Lotka in this class. Lotka was the mathematician that proposed the equation for logistic population growth that you're going to talk a lot about in this class. He also, a couple of years after this paper, published a paper on predator-prey dynamics that showed that predators and prey cycle in relation to one another. So why you get, it was the first model to sort of describe the dynamics that would give you cycling of abundances of predator and prey in response to one another. He also published a series of papers on models of competition for food between resource competitors. So Lotka's name is going to come up a lot in this class. But the first paper that he published of any note was an analysis of a model, this model that Ross had proposed the year before about humans interacting with malaria parasites. Now, Ross's and Lotka's models are, as I said, they're models of the interaction between two species, humans and Plasmodium, but Plasmodium's abundance doesn't enter into the model equations that Ross or Lotka used. So when we think about the models that you're going to talk about in this class, we're usually talking about a model that describes how the change in abundance of a species over time. Does the species increase in abundance? Does it decrease in abundance, or does it stay constant in abundance over time? And the equations, the models you're going to talk about in this class describe the dynamics of those processes and the changes in the abundances of those species. But the models that Ross and Lotka used did not include explicit equations to describe how Plasmodium abundances change. What they did was they built a model that said Plasmodium is there implicitly because we're going to follow human individuals that are infected with Plasmodium. So really the human becomes the surrogate for Plasmodium in the model. And this model that they worked on in 1911 and 1912 really is the progenitor of all the models that we use today to study the disease dynamics in host species. And you're going to see these as acronyms. You're going to see this acronym a lot today, and you probably hear a lot about it in the news. It's called the SIR model. Now, it's called the SIR model because what it does is it takes the host population, in this case, let's think about humans, and it partitions humans into three categories: susceptible individuals, infected individuals, and recovered individuals. And we're going to follow the abundances of these three classes of hosts. So what we'll start out with is a box that we'll call, this is the box that has all the susceptible individuals in them. So the initial population of hosts start as all susceptible. And we're going to assume that a pathogen jumps from another species into the host species we're talking about here. So these are what are known as zoonotic diseases. And a zoonotic disease is a pathogen that jumps from one species to another. And lots of infectious diseases in humans and other species are zoonotic. So flu comes from swine and birds, where the virus jumps from a pig into a human, and then that human starts passing the virus to other humans. HIV is another classic example. Ebola is another classic example. COVID-19 is a classic example now that we're dealing with. It jumped from some animal, we think either bats or pangolins, in China onto a human, and then started circulating in the human population. Now, all the models we're going to talk about in this class are really flows between these categories. So today we're going to talk about the flow of individuals between susceptible to infected individuals. So now we need another box to hold all the infected individuals. So when a susceptible individual becomes infected with the pathogen, that individual is transferred into the infected box. And this occurs at a rate that we'll call beta. So beta is just a number that describes the rate at which susceptible individuals are converted into infected individuals. And the dynamics of this are described by a model, which is a model component, which is known as a mass action process. So this really comes from, mass action really comes from physics. If you think about billiard balls rolling around at random on a pool table, every time two billiard balls clap into one another, an interaction can occur. In this case, a susceptible individual has to run into and interact with an infected individual in some way. Now, every time a susceptible and infected individual interact with one another, there's some rate at which that susceptible individual is converted into an infected individual. And so mass action is typically described as just the product of the rate at which this process occurs when these two individuals come into contact times the product of their abundances. So let me help you reason this out a little bit. So think about this term. This describes the total rate of movement of individuals from this bucket to this bucket. Now, why is it a product? Let's say there are no infected individuals in the population. Can any individual host in the susceptible category become infected if there are no infected individuals? Answer is no. So that would mean that the infected abundance is zero. So this would be zero. So there are no infections that are created when there are no infected individuals in the population. Likewise, if there are no susceptible individuals in the population, S would be zero. And so you could not have the conversion of a susceptible individual into an infected individual. Right? Everybody see that? You can't answer me, but hopefully you see it. Right? And so this is the way mass action works. So you'll see this again when we talk about predator-prey interactions. In predator-prey interactions, we have the abundance of the predator, the abundance of the prey. The interaction, the rate at which predators kill prey is actually given by exactly the same kind of term in the mathematics. Now, to complete the model, what we need are infected individuals to turn into recovered individuals. So that's why it's called an SIR model, SIR. Once individuals that are infected clear the infection, they become recovered individuals. And this occurs at rate gamma. So that's the Greek letter gamma. Now, this kind of model can only occur in species that have acquired immunity, right? We have an immune system where we produce antibodies in response to foreign invaders into our body. Once we develop antibodies against that type of invader, we now have resistance against that invader. And so we can fight off infections very quickly once we develop that immunity. So this will only happen in vertebrate animals and a few other groups that have acquired immunity. If you're in a group that does not have acquired immunity, we'd have to build a different model. But let's work with something that would work for humans here. This is the classic SIR model. And so now the rate at which individuals that are infected become recovered is the total rate is gamma times the number of infected individuals, because the rate at which recovered individuals emerge does not depend on the abundance of recovered individuals. So now we have our two main flows through this system, right? And all the models you're going to be talking about in this class are basically flow diagrams like this, where we're keeping track of we want to keep track of the number of individuals in each of these classes, and we're describing how individuals flow between these classes in the model. Now, in this model, we are not keeping track of the absolute abundances of individuals in each box. SIR models are typically built so that what you keep track of is the frequency of individuals in the buckets. And so we're going to assume that individuals, for now, the total population size stays the same. And all you do is move individuals between these three classes. And so that means that S, I, and R have to sum to 1, because that would account for all the individuals in the population, because it accounts for the frequency of all the classes in the population. So keep that in mind for a second, for the first part of this lecture, that we're really talking about the frequency of individuals in these three categories. Now, the way we describe the dynamics of movement between these boxes is with a series of coupled differential equations. Now, as I tell people in all the classes I teach, most people get very scared at this point for no good reason. And I ask students two questions. The first question is, did you pass 7th grade algebra? If you can answer yes to that, you have almost all the mathematical skills you need to play with these models. The second question is, do you understand what a derivative is? Do you understand what ds over dt actually means? You usually have to take the first semester calculus to understand that. But if you understand what a derivative is, and you can do 7th grade algebra, you will have no trouble whatsoever in this class, because all the models are very simple. You're not actually going to be taking derivatives or integrating functions. We're basically going to figure out how to write the equation down and then do some very simple algebra to play around with things to get answers. So let's think of these three equations. So remember, beta is the transmission rate of susceptible individuals into infected individuals. So you can think of beta as the rate at which when an interaction between a susceptible and an infected individual occurs, the infection is actually transferred from the affected to the susceptible individuals, right? That's because sometimes a susceptible and an infected individual will interact and the susceptible individual will not be infected. So the rate of change of susceptible individuals, that's literally what ds dt means, is the change in susceptible individuals with respect to time is simply minus beta si. Now it's minus beta si because when a susceptible individual gets infected, they move from the S to the I class, which decreases the frequency of susceptible individuals. So you're losing individuals from the susceptible pool. That's why this term is negative. Now those individuals go into the infected class, and so there's a term in the infected class rate of change that is exactly opposite of that. So it's positive beta Si, right? And that's simply because individuals leave here and move to here. And the same number that leave here move into this category. So these two terms are symmetrical. This is the equation, the total equation, that describes the change in the frequency of infected individuals with time. Now, you lose infected individuals from this pool because they move into the recovered pool as they move through. Right? And so we have a negative gamma I term here in this equation, and the description of the frequency of recovered individuals change over time is simply gaining those individuals into the recovered class. Right? So these are the three coupled differential equations that drive this SIR model. And this is the basic SIR model that most epidemiologists start with when they're talking about disease dynamics in a population of hosts. So what these equations describe is how this population of hosts changes as the infection proceeds through the population. And what we're doing is for this simple model, we're assuming that individual host individuals are not lost from the population. That's not a good assumption, but it actually does pretty well for what we're trying to describe. And remember, these are frequencies and not absolute abundances anyway. So if we lost or gained individuals from the host population, we just divide by a different total number of individuals to get the frequencies in these three classes. So if you look at the dynamics of this model over time, you'd map out this kind of relationship. So in this figure, the frequency of susceptible individuals is in blue, infected individuals are in red, and recovered individuals are in green. And those three differential equations in the previous slide actually give us what the shape of these curves are. Now, this figure I found online actually gives you the number of people in this model, but this really should be frequency. So we should divide by all these numbers by 1,000, and the maximum should be a 1, and the minimum should be a 0. But be that as it may. Right, so remember we said we start with all susceptible individuals, and then this pathogen jumps into this population of susceptible hosts and starts being passed around between hosts. And so now the frequency of infected individuals goes up, and the frequency of infected individuals increases, and the frequency of susceptible individuals decreases. But along the way, infected individuals then start turning into recovered individuals. And so what you see in this model is over the time course of the infection in this population, infected individuals have a peak. Now, epidemiologists use this model to try to understand the dynamics of diseases in host populations. So for example, here is data. So these are data, these little stars are data of individuals that were attending an English boarding school in 1978 when influenza swept through the population of students at this boarding school. So this is the number of individuals that were infected with flu over 14 weeks in this school. And what the people who wrote this paper that you can get down here by clicking on the link did was they fit this SIR model we've just been talking about to these data to try to estimate, get the best estimates for beta, the transmission rate of the flu virus between individuals in this boarding school, and gamma, the recovery rate of individuals going into the recovered individual pool. And they came up, after fitting this model to these data, they got estimates of beta of 0.5 and gamma of 0.0026. Now, that's what demographers and epidemiologists do with disease dynamics all the time. You can also use these models to project into the future, into project into the dynamics of the disease in the future. So for example, let's talk about a little bit, go back to the model for a minute and talk about what are the key parameters of this that describe the dynamics of this flow. So one of the most important dynamics in this flow is something called R naught. R naught is the ratio of the transmission rate between individuals, that number, and the recovery rate of individuals transitioning from infected to recovered, acquiring immunity in the population. This is the key metric of this model. And what it's called is the reproductive ratio of the pathogen in the host population. So it's basically a measure of how fast the pathogen is reproducing to create new infected hosts. Now, this number is also almost approximately the number of new infections that occur per infected host. So if this R naught was 2, that would mean that every infected individual causes an infection in 2 other host individuals in the population. So as R naught gets bigger, each infected individual is causing infections in more hosts. Okay, is that clear? Hope so. Now, this number is important because we can use it to understand whether the dynamics of this population, how the dynamics are going to occur in this population. If R naught is greater than 0, the pathogen will sweep through the host population. Now that's very important. So if we can get estimates of beta and gamma, we can make a prediction about whether this pathogen is going to be able to infect this host and persist for a while. Now, why is it that R naught being greater than 1 is the metric for this? So think about this for a second. Right? So R naught is the ratio of beta to gamma. Beta is the transmission rate from susceptible to infected, and gamma is the transition rate from infected to recovered. So if beta is less than gamma, that means individuals are moving faster from the infected to recovered class than individuals are in moving from the susceptible to the infected class, which means that you're decreasing the frequency of infected individuals over time because you've got more going out of that box than you've got coming into it, right? That's why that's what is required for R naught to be greater than 1 for the pathogen to run through the host population. Now, the other thing this implies is that the frequency of susceptible individuals at the beginning of the infection in the host has to be greater than 1 over R naught. This is implied by this relationship. So why is that? So you can see that by looking at the equation for infected individuals. So remember, we described this equation as the rate of change of the proportion of infected individuals in the population over time. Now, if that number is positive, that means that the frequency of infected individuals is increasing. If it's negative, that means the frequency of infected individuals is declining. Right? So when we start the population, we'd say, okay, susceptible individuals are really low in frequency. But if we ask the question, when is this total equation greater than 0, meaning that the frequency of infected individuals will increase from when they're initially introduced into the population, we basically are asking the question of when is di over dt greater than 0? So get a piece of paper and a pencil and just write this out on your paper and simplify it, right? So the i's cancel out, divide through by I, and we're left with beta s minus gamma is greater than 0. And then solve for gamma. If you solve for gamma, you end up with this relationship here. The frequency of susceptible individuals has to be greater than gamma over beta. But gamma over beta is just equal to 1 over R naught, right? So we end up with this relationship of the frequency of susceptible individuals has to be greater than 1 over R naught. Well, before we said that R naught has to be greater than 1 for the pathogen to sweep through the host population. So 1 over 1 means that the frequency of susceptibles has to be greater than 1, or greater than a number that's a really little smaller than 1, right? Because R naught is going to be a little bigger than 1. Now, the converse of this is that if R naught is less than 1, the pathogen will not sweep through the population. So basically, the pathogen will do what epidemiologists call epidemic burnout. So it'll infect the host population, but the infection will peter out itself, not because it sweeps through the population and forces all susceptible individuals to become recovered individuals, but because of simply not having a high enough transmission rate relative to the recovery rate for it to maintain itself in the host population. Now, as I said, R naught is actually important if it's above 1, and the magnitude of it being above 1 is also really important, because R naught is basically a measure of how fast the pathogen sweeps through the host population. So I'm sure you've heard a lot about flattening the curve over the last month or so. That has to do directly with R naught. So when R naught is very high, is a large number, this increase in infected individuals occurs very rapidly and dies off really quickly. Now, the problem is that creates for our healthcare system is that if there are more individuals than the healthcare system can deal with, a lot of people are not going to get healthcare. So by flattening the curve, what you are trying to do is lower the value of R naught so that the maximum as this pathogen sweeps through the host population, it does not overwhelm the healthcare system. Right? So let's think a little bit about what we're doing with all the things the CDC is recommending to flatten the curve. How does it change R naught? Right? How does hand washing decrease R naught? Is it changing beta or is it changing gamma? What about cleaning surfaces or social distancing? Right? The subject of those 2 papers we were talking about. And the reason you're watching this on a video and not sitting in a classroom with me, right? All 3 of these things are changing beta, and they're all trying to decrease beta. And if you can decrease beta, you decrease R naught, right? Hand washing decreases the probability that if you have received viruses on your hands, will transfer them to your nose or mouth and infect yourself. It also decreases the chances that you will pass on the pathogen to someone else if you have it on your hands. The same for cleaning surfaces. And social distancing, decreasing, as those papers show, decreasing the rate of transmission by decreasing the probability that individuals will be infected when they come in contact with one another, but also just decreasing the rate at which S bumps into I, right? Now, we can also do things to change gamma. Not a lot right now, but gamma can be changed, right? When you come up with medical therapies that increase the recovery rate of individuals, that decreases R naught. And if the host population has a stronger immune system to start with, recovery rate will occur faster, and so R naught will be smaller. So this is how we use all of these models to actually think about creating public policy. So people at the CDC, epidemiologists, are using this model right now, right? This is the basis of all the things that you're hearing from CDC scientists and scientists around the world because of analysis of this model. If we can decrease R naught, we can decrease the transmission rate. Without this model, we wouldn't know what to do. This is why scientists use mathematical models and why mathematical models are so important in science, because it gives you a way of summarizing effects that you know occur, the mechanisms of interactions, and to project what the consequences of those mechanisms are. It also gives you the chance of say, if we played around with the various features of this model, how would it change the dynamics of the system, right? That's what we're doing by saying, if I decrease beta, I get a decrease in R naught, and if I get a decrease in R naught, the curve of infected individuals over time will look like this instead of like that. So that's what we're using. That's why mathematical models are so important to scientists. And it's why you should really push yourself to become proficient with using mathematical models. Now, let's add a little wrinkle into this model. Let's say that we add a vaccination program. So hopefully we'll get a vaccine relatively soon. What do vaccines do? Well, what vaccines do is they transfer susceptible individuals directly into the recovered class. If we have a vaccination rate of nu, so that is the Greek letter nu, we can describe the dynamics of individuals moving from susceptible to recovered by the rate at which we vaccinate susceptible individuals. So now we've got a third rate in this model. What this does is it changes the equations very slightly. So now we have excuse me, a second term in the susceptible category, or in the susceptible equation, that describes the rate at which you lose susceptible individuals to recovered individuals category, and those individuals are picked up in the recovered equation. So we haven't changed the infected equation at all, because nothing was changed there because of the vaccination program. The only thing that vaccinations are doing is transferring susceptible individuals into recovered individuals. Now, this is where this little equation becomes important, because we said S is always going to starts as 1 in the original model. But if you think about it, what vaccinations are doing is decreasing the frequency of susceptible individuals in the population. Right? And so what vaccines are doing is it's making it harder for pathogens to maintain themselves in the host population, where you have a high frequency of vaccinated individuals, because you've decreased this number. And so it's harder for this inequality to be true. So think about it. If R naught is 2, then 1 over R naught is a half, which means that the frequency of susceptible individuals, if they're less than a half of the population, this population is going to burn out. This pathogen is going to burn out. If R naught's 4, that means that the frequency of susceptible individuals has to be greater than 1/4 to prevent. But if the frequency of susceptible individuals is less than 1/4, that pathogen is going to burn out and not sweep through the host population. So that's what vaccinations do. And that's how we deal with vaccinations in this model. Because what we're trying to create is something known as herd immunity. So herd immunity is where so many individuals in the population are immune to the disease that the pathogen is always going to burn out. And this occurs, we can analyze a more sophisticated version of this model, and you come up with the result that the frequency of vaccinated individuals has to be greater than 1 minus 1 over R naught. So if R naught is 4, 1 over R naught is 1/4, and that means that more than 3/4 of the population have to be vaccinated for herd immunity to be effective. Right? This is how we eradicated smallpox in the human population. That is why smallpox is now extinct in the human population. It's also why chickenpox and measles and rubella are becoming a problem again, because people aren't vaccinating their kids against these childhood diseases, and now they're becoming a problem. You're seeing a much greater prevalence of them worldwide in the last decade. Now let's end this by talking about, let's build in this model. We'll build in a couple more complications into this model. So you can see the full scope of this. And this is how we develop models in science. You start with a very simple model, understand its dynamics, and then you start adding pieces to it. So we started with the very simple SIR model. That's the simplest one that we have to play with. Even that very simple model makes very good predictions for a lot of things we want to understand. But then we can add in new features. We can play around with saying, if we introduce vaccination into this problem, what has to be true about the rate of vaccination to have a substantial difference on the dynamics of the system? That's why we use models. We can play around with, without actually having to do it, we can test out hypotheses and scenarios to see if the problem will be satisfied, so we can test it out there before we have to actually spend a lot of money to actually do something and see if it works. So the model really becomes the proving grounds for whether ideas are going to potentially work or not. If the model is reflective of the mechanisms operating, more than likely they will give you predictions that will be borne out when you start doing empirical studies. That's what we're doing here. That's why we're dealing with models, and that's why you're going to play with models a lot in this class. Now, let's add another feature to this. So let's make this a little more realistic by saying that individuals actually have offspring, so individuals are born, and individuals die. And we'll include the dynamics of birth and death into this model, so that will increase its mechanistic description of the demography of this population. So demography is the description of changes in abundance by the mechanisms of birth, death, immigration, and emigration into the in and out of the population. So the first thing we need to do is change the way we think about the state variables in this model. So now S, I, and R are not proportions anymore. They're actually absolute abundances. So we're going to keep track of, there are S individuals, and that means there's 862 S individuals and 137 I individuals and 47,000 recovered individuals, right? So these are numbers that are not between 0 and 1, not proportions, but they're absolute numbers of individuals in the population that we're keeping track of the host. But all the same logic applies in what we're going to work through, so that we don't have to change a lot. So first of all, we're going to say that we're going to make the assumption that susceptible individuals have some birth rate that produce new susceptible individuals in the population, right? And we'll call that rate B. Susceptible individuals also have some death rate. They get old, they die of other causes, so there's some natural background death rate for susceptible individuals, and we'll call that D times S. And this is going to give us the ability to regulate the abundance of this population. And you'll talk about this more in future lectures in this class, because this is generating population regulation. So this population will not increase to an infinite population size. It will stabilize at some number of individuals because of birth rate and death rate being this so. Wait. We'll also assume that infected individuals have a death rate, but the death rate of infected individuals is higher than the death rate of susceptible individuals. And it's higher by a factor of theta. So we're going to multiply D times theta to get a higher death rate for infected individuals. And so what we're assuming is theta is greater than 1. So D is the per capita death rate of susceptible individuals. Theta times D is the per capita death rate, the rate at which an individual will die for infected individuals. So now this disease increases the death rate in this host population, but it only increases the death rate for individuals that are actually infected with the host. If they recover, that individual's death rate drops back down to D times the number of recovered individuals in the population. Right? Okay? I hope everybody's on this. And so we have the same transition rates between these three classes, but now you don't really have to pay attention to this unless you really want to. But if you think about it, beta and gamma now have slightly different interpretations because we're not talking about transitions between proportions. We're talking about transitions between absolute abundances. But the same logic applies, as I said. So now we can write down exactly the same equations, but now remember, we don't have vaccinations in here. So this component of the model for susceptible individuals is what would happen in this population were there no infections, were there no pathogen. If this pathogen was absent from the population, this equation, this part of the equation describes the change in abundance for the susceptible individuals. And so this population would be regulated actually at an abundance where S is equal to B over D. But you'll learn about that later in the class. But it also has this loss of individuals from transitioning from susceptible to infected, just like we had in the original model. In fact, the infected individual equation for the change in infected individuals per time is exactly the same as we were talking about before, except now we have a death term where you lose individuals from the infected class. And then the same thing for recovered individuals. We lose some recovered individuals per unit time because of death. Now, once we analyze this model, it doesn't change things all that much. What it does change is it changes what we would calculate as R naught slightly. And R naught, remember when we were talking about proportions, was just beta over gamma. But now R naught has this added death term of infected individuals. And you could actually calculate this by taking again the equation for the infected individuals, setting it equal to 0, and solving for 1 over S. You actually get this, right? So now R naught is a function of both transmission, recovery, but also death due to being infected. So the death rate of an individual due to the infection enters into R naught. So let's think about this a little bit. What does this mean? So now epidemics can burn out for two reasons. One of them can be that gamma is greater than beta, the same reason we had before. Now, I'm sorry, let's step back a second. So now, before we analyze this, remember we were talking about proportions, and we had the proportion of susceptible individuals has to be greater than 1 over R naught. Well, because we're talking about absolute abundances now, we have to divide the absolute abundance of susceptible individuals by the total population size, which is just, we'll call N. And so the total population size is just the sum of all the individuals in these three categories. So when we divide S by N, we get the proportion of individuals in the population that are susceptible. And so we're back to exactly the same formula we were talking about before in the previous model when we were talking about proportions. So now if we substitute our new R naught into this equation, we end up with this. We end up with the frequency of susceptible individuals has to be greater than this quantity, or this quotient. Now let's think about this. So if in this model, right, gamma and beta are still the same problems, but death of infected individuals makes it harder for the pathogen to maintain itself in the host and to sweep through the host. And in fact, the greater the death rate caused by the pathogen, the harder it is for the pathogen to maintain itself. Right? This is actually why Ebola typically burns out when it infects human populations. So Ebola pops up periodically in small human populations in Africa, because that's where the Ebola is endemic. When it jumps from its secondary host, which is not a human, into the human population, and then starts spreading in humans, the death rate from Ebola is so high that the Ebola typically burns out in those local populations. So think about this. What has to be true of an individual that's infected to infect another individual? They have to live long enough to run into a susceptible individual. And so if you decrease the time that an infected individual is alive, you decrease the chances of passing that infection on to another susceptible individual. This is exactly the same thing, exactly the same idea about quarantining, right? You put individuals into quarantine to decrease the frequency of them passing on the pathogen to another host or to a susceptible host. So quarantining is actually simulating the death of that host until that individual becomes recovered. So quarantining also plays the same role as this term in this model. That's why we impose quarantine on some. This is the justification for it, because it drops out of these models to say if you quarantine individuals, you're making this ratio harder for the pathogen to beat, and so the pathogen will hopefully die out in the host population. So why was the Ebola outbreak of 2014 and 2016 so different from previous outbreaks? Now we're going to talk about this a little bit, because these are the kind of questions you're going to get on the exams in this class. And we want you to be able to translate ideas about real-life situations into models and take model predictions and translate them into real life. So Ebola was extremely deadly, is an extremely deadly disease. It typically burns out. But in 2014 and 2016, it took a very, very, very long time for it to burn out. Why did it not almost immediately burn out? Well, it did not almost immediately burn out because it got into a few cities in Africa where the transmission rate, beta, went way up. So because you're in a different human population, where individuals are packed together, beta becomes very big. So now you've made this quotient a much smaller number. And so now it's easier for the disease to beat that ratio. That's why it took Ebola so much longer to burn itself out in the 2014-2016 outbreak. Right? So here's another example of how we can use these models to actually understand the dynamics in real populations. Right? So here's another question to think about. Given we have this inequality, why do flu and the common cold virus continually circulate in the human population, but Ebola burns out? What is different between these different types of pathogens that cause this different type of dynamic between these various infectious diseases? That's exactly what scientists at the CDC work with every day. That's exactly what scientists around the world who do epidemiology of infectious diseases are thinking about every day. They also are building more complicated sets of these models to try to make even more precise predictions about how the dynamics of various diseases will go, and to actually try out different public policy proposals to see how effective they might be. Because you can't, right? Think about public policy. You have to project into the future. You have to make a prediction about what will happen when you do something before you've ever done it before. Because you don't usually, for a new public policy, have the ability to take little test populations and try it out and see what will work and then go back and do it. Right? Particularly with something like this, you've got to just make your best prediction with the best evidence you have and use that as a starting point. That is what CDC scientists and epidemiologists around the world do with these models, is they know they've been effective at predicting the dynamics of other diseases. So now if we can make educated guesses about the magnitudes of these various parameters, we can understand what we need to do to deal with new infections. That's what we're doing right now. So I hope you've enjoyed this. I hope you've learned something. So now what I want you to do is to go back and read these two papers again. Think about what the papers were talking about now, given your new understanding of SIR models. And think about what sorts of new public policies may be effective in dealing with infectious diseases. Thank you for listening. And if you have any questions, I'm sure we'll be talking on Canvas. Thanks. Talk to you later.